Okay, students, so welcome back again. This is your third part of microbes in human welfare. Okay, in this part, we are going to discuss about biocontrol and how that biogas is produced by microbe, which is utilized by us. Okay, so first we have to know from where, what organisms, what microbes are responsible for the formation of this biogas. Okay, so in 11th standard, you studied about biological classification. You studied the chapter biological classification, in which you studied a category of monerans, that is archaebacteria, in which you studied three types of bacteria, that is thermoacidophils, who is live in very high temperature areas. Second one is halophiles, which live in highly salinity areas, and third one is your methanogens, who lives in marshy area. So those methanogens which you studied in biological classification, they are here again in 12th class. That is your methanobacter, methanococcus, methanococci. All these are different types of methane producing bacteria. So where they are going to produce that thing, where they are present now? Okay, they are present inside the gut of cattle. Okay, you will find these bacteria inside the gut of the cattle for the digestion of cellulose. Okay, so same thing is written. Methogens are the bacteria in the rumen of the cattle. Rumen is the part of the gut of the cattle. Okay, that's why we call them ruminants. Okay, ruminants are those animals whose gut contain rumen. Okay, so these bacteria help in digestion of cellulose. Okay, these bacteria methanogens are responsible of breakdown of cellulose inside the gut of the cow, and when that breakdown of cellulose take place, you get one gas, which is your methane. Okay, which is the main constituent of biogas. Okay, so methane are also found in the cow dung. Okay, methanogens are also found in cow dung. Okay, as you know, cow dung, it is the excreta of cow. Okay, so the same thing is coming out from your digestive tract. Automatically that cow dung contain this methanogens. Okay, the most common methanogen is methanobacterium. Okay, it is the most commonest methanogen which is present now in the world, that is methanobacterium. In the rumen of cow, you will find methanococcus and rumenococcus. Okay, these two type of methanogens are present inside the gut of cow. Again, I am saying the name, that is methanococcus and rumenococcus. Okay, so this is the cow dung with methanogen, and this is your methanobacterium, which is present inside the cow dung. Okay, so cow dung contains cellulose in it. Okay, as you know, cellulose does not get digested fully. Some part of cellulose, if it is 100%, then 10% part of that cellulose which is eaten by cow. Okay, 100% eaten by cow, 10% of that 100% does not get digested. So it come out with excreta. Okay, so methanogen present in the cow dung act on that cellulosic material anaerobically. Okay, again I am saying this process of production of biogas is anaerobic process. Okay, there is no use of oxygen, it is purely anaerobic process. No oxygen is there. Okay, so they result in production of biogas. Therefore cow dung is widely used in the production of biogas, also known as gobar gas. Okay, as you know, the excreta of cow is called gobar, the cow dung is called gobar. Okay, that's why the gas which is produced by this cow dung, this gobar, is called gobar gas, or your other name of your biogas is gobar gas. Okay, so this cow dung with methanogen, anaerobic respiration done, and you will get biogas or your gobar gas. Okay, so what is this biogas? What is the biogas? So biogas is not a single gas. It contains different gases. It is a mixture of different gases like methane. Okay, hydrogen sulfide, hydrogen, CO2. Okay, so biogas is a mixture of different gases that are produced due to the microbial activity of methanogens. Methanogens act anaerobically on your cellulosic material, and when that cellulosic material break down in anaerobic condition, it produce biogas. Okay, so it predominantly consists methane, appropriate 50 to 70%. Out of 100% of biogas, 70% is methane, and that methane is the combustible thing. Okay, the fire which biogas caught, it is because of methane, because methane is a combustible gas. It can easily get fired. Okay, so carbon dioxide, hydrogen, and few other gases present in the biogas, that is in trace amount. Okay, so what are the components of biogas? Mainly it is formed by methane, CH4. Okay, then you have CO2, hydrogen sulfide. These all are other gases which is present in biogas, but it is like least in number. The prominent number, the highly prominent number which is present inside this is your methane, that is 70%. Okay, so you can see here, okay, biogas, it is an diagrammatical showing you biogas in which you will see methane, everywhere methane. 70% area occupied by methane. Then your CO2, then hydrogen gas, and then in trace amount small gases like hydrogen sulfide and other gases. Okay, so but you have to remember one thing, that is you have to alter inside your brain, that you have to contest inside your brain that biogas is chiefly and predominantly made up of methane. Okay, next, now what are the uses of biogas? Why we are producing biogas in different areas, so we have some use for that biogas. Okay, so first, it can be used for cooking food and other domestic heating purpose. Okay, just like your LPG, CNG, petroleum, coal, which you use to cook the food, which you use as a heat source, this can biogas, it can also use as a heat source, it also can use as a food cooking material. Okay, by you can easily cook the food. Okay, it can be used for street lighting. Okay, in foreign countries like European countries like your Norway, okay, Sweden, Switzerland, they use this biogas to light up their street lights. Okay, next, it can be used as a fuel to run small engines like water pumping set for irrigation purpose. Okay, just like you use in the pumps diesel. Okay, water pump में diesel use करते हैं, उसकी जगह biogas use करो. Okay, biogas have very high calorific value. Okay, and it does not get that much polluted the environment. Okay, pollution is very less in the case of biogas. Okay, so these are the uses of biogas at last: cooking, lighting, and fuel. Okay, so now how you are producing biogas? What are the procedure behind the production of biogas? Okay, so the technology of biogas production was developed in India mainly due to the efforts of Indian Agriculture Research Institute IARI and Khadi and Village Industry Commission KVIC. Okay, in NEET exam they will ask you which government body in India giving funds and allowing to produce biogas. That is your Indian Agriculture Research Institute and Khadi and Village Industry Commission KVIC and IARI. These two government bodies are controlling the production of biogas on large scale in India. Okay, in a typical biogas plant there is slurry tank. Okay, here you can see. Okay, this is the slurry tank. In this tank you will put raw material, means cow dung. Okay, vegetable peels. Okay, cropland waste. Okay, sewage. Now in the treatment of water, and we studied that when we put that sewage in that area where that non-digester, that anaerobic digester, then the thing is send it to biogas plant and it is added inside the slurry tank. Okay, so the raw material for your biogas production are cow dung. Okay, household waste, vegetable waste. Okay, then your that sludge from your that water treatment plant. Okay, so all these are the raw material which you add inside the slurry tank. Okay, now from this slurry tank it will go inside a main digester. Okay, so this is your digester and it is covered by a cover which is your gas holder. Okay, now cover which act as a gas holder and sludge tank. Here now you the remaining the end thing. Okay, the end thing which is not going to be used has been stored here and it will after some time it will use as a biofertilizers. Okay, again I am saying this is your slurry tank where you add raw material and it will go inside the digester where the process, the anaerobic digestion of that raw material take place, and now from that anaerobic respiration your methane, that biogas, will come up and accumulate in gas holder. Okay, and when whole substance is digested anaerobically, then that thing is sent it to the sludge tank and then it is used as a biofertilizer. Okay, so it is zero waste product. Okay, you are not getting any waste. Even the waste which is produced is also used, okay, as a form of biofertilizer. Okay, so the digester is 10 to 15 feet deep in the soil. Okay, you have to put this digester deep in the soil. Okay, then you get that anaerobic condition. Simultaneously, there is chance that due to the over accumulation of gas, this can be blasted. So that's why we will dig a 10 to 15 feet deep hole, then we make this digester. Okay, so a slurry is made by mixing cow dung and water. Okay, this slurry can also contain organic waste, as I say, that peels. Okay, and that material which is coming from your water treatment plant. Okay, this slurry is poured into the slurry tank, and through the inlet it enters into the digester. As you seen here, this is the slurry tank which is contain green slurry. Okay, that product, and it will go inside the digester. Okay, so the digester is dug deep in the soil. It provides the perfect environment for microbes and respiration anaerobically. Okay, as I said, it is digged that much deep, 10 to 15 feet deep, because it have contain an aerobic condition. Okay, that deepness in 10 to 15, it will provide a better environment to your bacterium, to your methogen to do anaerobic respiration. Okay, then during anaerobic respiration of methanogen, the cellulose material is used as substrate to produce gas like methane, CO2 and hydrogen. Combined together, these are called biogas and gobar gas. Your bacterium methanogens, okay, they act on the cellulosic waste. They break down that cellulose into gases like methane, CO2, hydrogen sulfide, and after that this gas mix up and form a single gas which is your biogas, and that biogas is being accumulated into the gas holder. Okay, you will see here. The gas then pass through an outlet and is supplied to different household. Okay, now this is your gas holder which contain this gas, and this is the pipeline by which it will supply to different area. The sludge tank collect the overflow slurry in the digester. Okay, when sometimes this will fill up, then this will accumulated in sludge tank. Okay, since cattle sheds are mostly found in the rural area of India, biogas plant are also mostly seen in rural area. Okay, as you know, farmers, these all are belong to the rural area of India. Okay, farmers have the cow, they have their that go shala. Okay, that is present mostly in rural areas. Okay, so you will find biogas plant in rural area mostly. In urban area there is also, they are present, but not much. That it is mostly present in rural area because the raw material, the important material, cow dung, you will get exclusively in that farmer lands or in the rural area of India. Okay, now disadvantage. Now we are going towards biocontrol. Okay, that is your biogas production. Okay, how your biogas is produced? It is produced by with the help of methanogen bacterium act on raw material like cellulose, then the household waste and sludge which is coming from your water treatment plant, and they will acting on that and digesting them anaerobically and producing gases like methane, CO2, hydrogen sulfide and hydrogen, and these gases come together and forming a single gas which is your biogas or gobar gas. Okay, next we have going towards your thing, that is biocontrol. Okay, before starting biocontrol we have to know why we are going to apply biocontrol. We can use chemical control also, but why we are using biocontrol? Okay, so agrochemicals are too expensive for farmers to afford. Okay, the reason behind we are using biocontrol on the place of that chemical control on pest is agrochemicals are too expensive. Okay, the chemicals, the fertilizers, the pesticide which is produced chemically, which is composed of chemicals, totally chemicals, 100% chemicals, these are very costly. Okay, second, yield could not be increased further with existing variety using conventional method of agriculture. Okay, you cannot increase your yield by using that. At a time it is increased, but after some time it will not increase that much. Okay, agrochemical cause environmental and health related issues. Okay, that is the nowadays very prominent issue, that why we will eat the food which is sold in the market. Okay, the fruits, the vegetables, it is good for our health or not? Half of them are inserted, agreed by the help of inserting different chemicals. Okay, जो अभी सब्जियां फल मार्केट में आ रहे हैं वो हमारे लिए अच्छे हैं या नहीं, हमें कैसे पता लगेगा? ओके, उसके अंदर केमिकल भरे हुए हैं, पेस्टिसाइड का छिड़काव कर करके उन सब्जियों को और फलों को जहर बना दिया गया है. ओके, सो get out from that thing, we are start using biocontrol. We are using to decrease the pest inside the farmland by using biocontrol measures. Okay, so biocontrol agents and biofertilizers. Biocontrol agents means those agents which decrease and which delete and which make die the pest from your farmlands. Okay, while biofertilizers are those fertilizers which are added to the cropland to increases the soil fertility, to increases the power of the soil to give nutrients to the plants. Okay, so microbes which are used in controlling the plant diseases and pests are called biocontrol agents. Okay, biocontrol using microbes is based on the natural predation and not on the chemical. Okay, in this method you are using natural predation ecology. You are not disturbing the ecology, you are using a better way. If you add chemical inside the field, it will destroy the pest which you want, and additionally it is also destroy the organism which is living in your cropland. Okay, for example, if there is a cotton ball roller insect is there which is destroying your cotton field, if you put chemical pesticide inside the field, it will surely make die your cotton borer. Okay, but that chemical pesticide can also kill other organism which is living inside your cropland, just like earthworm, which is the farmer friend. Okay, so due to that thing we are going specific, we have to destroy the specific pest, specific organism which is only destroying the your crop. Okay, we do not want to destroy that organism which is good for you, which is does not harming your crop. Okay, and due to that thing you are balancing the ecology of your cropland. Your cropland is also ecosystem. There is abiotic forces, light, sunlight, okay, soil, air, water, temperature, and biotic things are also there, different types of insects, microorganisms. Okay, if you want to kill a single species, then go specific. Okay, and how can you go specific? By using biocontrol. Okay, by using biocontrol you can go specific. Okay, it is one of the important part of integrated pest management, IPM, integrated pest management, in which you use different techniques to cope up with the biodiversity, to do not destroy the biodiversity of your croplands. Okay, so what is your integrated pest management? Okay, so integrated pest management approach is a very economical. Okay, it is very economical, it is not much that costlier, just like your orthodox chemical fertilizers. Okay, baculovirus is considered as a desirable factor for integrated pest management as it helps in controlling pests without affecting the species. It is specific species killing organism. If you want to kill the organism which is only destroying your crop, you will go with baculovirus. Okay, so integrated pest management, IPM, knowledge of pests, their life cycle, their interaction with the environment help in developing a holistic approach in managing the pest. Okay, in integrated pest management you will go and know what is the relationship between the host and parasite inside your crop field, how that the life cycle of the pest runs inside your cropland, and how can you eliminate that pest without harming other organism, other that plants or animals which is not doing any damage to your crops. Okay, that thing is come under integrated pest management. Okay, so we have three advantage of using microbes as a pest control. Okay, first, do not cause pollution to the environment. Okay, automatically it is a biological thing, it is a bio thing, it is a organic thing, so it does not do any damage, any pollution to that particular ecosystem. If you use chemical pesticides, chemical agents who do biocontrol, it will definitely pollute the area. When it goes inside the soil, it will go do soil pollution. When it will sprinkle in the air, it will do air pollution. When it will go and mix with the water which is giving for the irrigation of the field, it will pollute the water also. So your pesticides, chemical pesticide is doing soil pollution, air pollution, and your water pollution. But your biocontrol agent, the biological, the organic pesticides, they only go and kill the pest which you want, which you do not want inside your field. Okay, so by using biocontrol, okay, that using organic pesticides, you will avoiding pollution inside your own ecosystem of your farm. Okay. Second, in biocontrol, beneficial pests are retained but harmful pests are not. Okay. Beneficial pest means some organisms is beneficial, okay, to your cropland. Some are not, which will you want to destroy. So by the help of biocontrol, you are going specific. You are not a narrow, that broad. Okay, you are not killing the broad killing you are doing. By using biocontrol, you are killing only the specific organism that you do not want inside your crop field. Okay, and this is your base on predation system. Okay, the predator you introduce inside the cropland of that organism, it will eat only that thing, it will not touch other organism. Okay, and the third, that advantage of using biocontrol, okay, does not eradicate pests but keep them under management level. Okay, your biocontrol measure is not going to totally eradicate that pest. Okay, it will manage the number of pests inside the cropland. Okay, see, integrated pest management does not allow you to remove all the pests from the cropland, because that pest is also the part of ecosystem. If we remove these organisms from that ecosystem will become imbalanced. So by using biocontrol agent, you are doing what? You are controlling the ecosystem. You are not making that ecosystem disbalance. Okay, so hence the ecosystem is not disturbed. Okay, if you do all the pests get killed, then automatically the ecosystem get altered. Okay, it get decreased, it get affected, it get disturbed. But when you go with biocontrol agents, which is specific but it is killing in a normal number, not that extensive killing, all killing, then your ecosystem is not disturbed, it is on a proper pace. Okay, and this thing, the consumption of chemicals sprayed on vegetable and fruit for long period can result in certain health issues. Okay, as you know, I said previously today that today's worst concern about eating a fruit and vegetable from market is that either it is pesticide free or not. We do not know either that fruit or vegetable which we eating in the dinner or in the lunch or in the morning in the breakfast, it is pesticide free or not. So how can you do that thing chemical pesticide free? By adding and by using biocontrol agent, by using organic pesticides. Okay. So we have some examples of biocontrol agents. First we have Bt cotton. Okay, Bacillus thuringiensis cotton. Okay, so the gene of Bacillus bacteria is inserted into the plant. We are going to study that thing in detail in biotech in its application, where I will tell you how we introduce that Bt gene inside the cry gene of Bacillus thuringiensis inside the cotton plant. Okay, now you have only remembered the name of the organism and how it works. Okay, so the gene of Bacillus bacteria is inserted into the plant. Okay, this gene is called cry gene, the gene which is responsible for the production of Bt toxin inside that crop is called cry gene, CRY cry gene. Okay, so the gene is called cry gene as it is code for crystalline protein. Okay, the full form of a cry gene is crystalline protein, which is a toxin. Okay, it is a toxin to the insects like cotton borer. Okay, cotton bollworms. Okay, cotton army worms. These all are the pests to your cotton fields. So these are the poison. These are the toxin for these insects, these pests. When they eat that, their gut get destroyed. Okay, this toxin is produced in inactive form. Okay, ये inactive form में produce होता है, but जब ये insect की gut में जाता है, ये activate हो जाता है. Okay, these toxins are produced in inactive form, but when these toxins get inside the gut of that pest only, it get activated while getting that alkaline pH of the gut. Okay, means this toxin required alkaline pH, and that thing is present inside the gut of that insect. Okay, and when that insect eat this, the plant part which will incorporated with this toxin, when that insect eat that, it automatically go inside the gut of insect, and that toxin get activated under that alkaline pH, and insect get killed. Okay. So when the worm feeds on the Bt bacteria, due to the alkaline medium of insect gut, protoxin get activated. Your Bt protoxin is get activated under that, and it will activate, it will start to destroy the gut of your insect. Okay, the activated crystalline protein toxin then solubilize in the warm gut and kill it by doing perforation in the gut. Okay, see, inactive Bt protoxin. Okay, now it will eaten by your insect larva. Now the larva, the warm gut having alkaline pH. This alkaline pH activates your Bt toxin, and now that die. Okay, RIP, khatam, tata, rappa rappa. Okay. Next example is fungi as a biocontrol. Previously it is your bacterium. Now we are going to talk about fungus. Okay, that is your trichoderma. Okay, this trichoderma fungus live inside the roots of the plants. Okay, trichoderma is a species of fungus found in the root ecosystem of the plants. It controls several pathogens affecting plant and act as a biocontrol agent. This fungus inhibit the entry of microorganism and paste inside the body of tree while the way of रूट से जो इंसेक्ट्स और पैथोजेंस अंदर आते हैं प्लांट के उसको रोकती है ये फंगस ट्राइकोडर्मा. ओके, ट्राइकोडर्मा स्टॉप दैट थिंग, इट विल इनहिबिट्स द एंट्री ऑफ दैट थिंग. ओके, okay, it will inhibits the entry of pathogens inside the plant body which enters by root because it grow inside the root. Okay, so this is your trichoderma pathogenic fungus. Okay, identify and attack plant pathogen fungus. Okay, so nematode trichoderma destroy nematode eggs. Okay, this fungus destroy nematode eggs. Now your nematode cannot go inside the plant body and affect the plant. Okay, plant root pathogen fungus, it will bind with the roots. Okay, induce resistance to the plant. Okay, just if this is your root. Okay, and this is the fungus, it will come and go and bind just like this. Okay, now it will make a circle around your root, and now no pathogen have the power to get inside. It have to first fight with that fungus, and in that time it will die. Okay, due to that toxin which is released by this fungus trichoderma. Okay, now virus. Okay, first we studied bacterium, that is Bt, Bacillus thuringiensis. Second we studied a fungus, that is trichoderma, and third one is now virus. Okay, so virus we use baculovirus. Okay, baculovirus is a pathogen that attacks arthropods. Okay, these viruses belong to the genus Nucleopolyhedrovirus. Okay, baculovirus is the virus, and the genus from which this baculovirus belong, it is Nucleopolyhedrovirus. Okay, Nucleopolyhedrovirus is the genus from which your baculovirus belongs. Okay, so Nucleopolyhedrovirus genus, your baculovirus, it kills insects and other arthropods which want to enter inside the plant body to destroy the plant body. Okay, so this is again the third way of using a bioorganic thing to control the pest. Okay, so how this virus act? Okay, so the specialty of this virus is that this is resistant to the host range and does not kill other organism. Okay, it is a very specific parasite killing organism. Okay, it only kills a single species at a time. It does not go and kill other organism at a time. It is a very specific organism which is used as a biocontrol. Okay, so it does not affect vertebrates, non-arthropod, invertebrates, microorganism or plants. Okay, it is not going to affect these organisms. It only affect the organism which you want to kill. Okay, the organism, the pest which is inhibiting, which is avoiding, which is reducing the growth of your crops, it will killed by this virus. Not other organism of your crop field is killed by this virus. Okay, that's why it is very specific in killing. Okay, these viruses are excellent candidate for species specific narrow spectrum insecticidal application. Okay, narrow species means narrow spectrum species killing means it will only kill one species at a time. Okay, it will only kill the species you want to be eliminated from your cropland by the help of this virus. Okay, that's why it is called narrow spectrum insecticidal application virus. Okay, you can easily introduce this bacteria, this virus inside the field, and it will kill only a narrow spectrum organism that you want to eliminate from your field. Other organism of your field which is you do not want to kill, which is an important part of your ecosystem of your field, is not eliminated. Only the bad one, the bad guys are eliminated by this virus. Okay, they have been shown to have no negative impact on plant, mammal, bird, fish, or even non-target insects. Okay, as I said previously. Okay, so these are this is baculovirus. It does not affect on vertebrates, non-arthropod, invertebrates, and plant. It only acts on arthropods. It only kills arthropods. Non-arthro invertebrates is safe, vertebrates safe, and plants safe. Okay, again I am saying plants safe, non-arthropod invertebrates safe, vertebrates safe. Only killing is of arthropods. Okay. Then animal control. Okay, now the fourth control, the fourth way to do biocontrol. First we studied bacteria, that is your Bacillus thuringiensis. Second we studied about a fungus, which is your trichoderma. Third we studied about a virus, that is your baculovirus. Now the fourth is your animal. Okay, how can you use an animal, multicellular animal, to kill organisms? See, you have two animals, ladybird and dragonfly. Okay, these two arthropods, ladybird and dragonfly, is used as a biocontrol agent. Okay, ladybird is used to kill aphids. Okay, this is your ladybird. It eats the aphid larvae, and now this aphid does not sucks your plant stem sap. That sap, it is present on the sap. Okay, जो plant के sap को खाते हैं, stem पे बैठ के sap को खाते हैं, वो अब नहीं होगा. Okay, so it is the natural predator of aphids. In this technique of using animal, you are using the predation method. Okay, you are throwing the natural predator of that organism inside your crop field, which eating that paste. Okay, so now we are using ladybird, and this ladybird eats its prey, that is aphids. Okay, second we have dragonfly. Okay, dragonfly, it is a predator and its prey is mosquito and mosquito larva. Okay, so that's your dragonfly. It is also called as mosquitocidal insect. It eats mosquito, just like in animal kingdom you studied a fish, gambusia. Okay, that gambusia fish, it is responsible, it is used as a mosquitocidal. The areas where the high risk of malaria, dengue, chikungunya is there, where stagnant water is there, we introduce that gambusia fish, and this fish eats the larva present inside that water, stagnant water. Okay, secondly we have dragonfly. It is also used as a eating mosquito. It is also used as a mosquitocidal insect, mosquitocidal animal. Okay, it eat all the larvae of mosquito, and due to which it will get effects, reduce down the number of that mosquito from your field. Okay, so one way to provide nourishment to crop is through the use of fertilizer. Okay, now we are going to study about biofertilizer. Till now we studied about biopesticide, biocontrol of pest, in which we studied 4 ways. How can you do that thing by 4 ways? First one is by using a bacteria, Bacillus thuringiensis. Okay, second by a fungus, that is your trichoderma. Third by that virus, which is your baculovirus, and fourth by animals like dragonfly and ladybird. Now we are going to discuss about biofertilizers. How to increase the yield, how to increase the fertility of your soil. We use biofertilizer. We have 2 types of fertilizer. One is biofertilizer, one is chemical fertilizer. Chemical fertilizer increases the yield, but it definitely do water pollution, soil pollution, and after some time it will also damage the pH of the soil. The pH of your soil get damaged after 1 year. Okay, then you are also again, now you have to add that fertilizer again to increase the fertility of the soil. Okay, so the one way to provide nourishment to crop is through the fertilizer. Okay, you can use fertilizer increase the crop. Fertilizer mainly made up of ammonia. Okay, that is nitrogen rich compound. Okay, so it increase the nitrogen uptake of the plants. So these are chemicals that contain essential element for the plant growth. Okay, they are diverse group of products that contain nitrogen, phosphorus, potassium, and other trace minerals. Okay, these 3 elements, these three metals, nitrogen, phosphorus, potassium, these are the most important thing for a plant growth. If these are present in soil in abundant number, your plant can grow very fast and it have a healthy growth. Okay. However, use of chemical fertilizers result in increase in pollution. Okay, as you know, I said chemical fertilizer, they cause water pollution, they cause air pollution, they cause soil pollution. Okay. So how can we avoid this? By using biofertilizer, by doing organic farming. Okay. So first we have a bacteria which is act as a biofertilizer, that is your cyanobacteria, BGA, blue green algae. Okay. So microbes that enhance the soil nutrients quality of the soil. Biofertilizers are the microbes that enhance the nutrient quality of the soil. The use of microbes instead of chemical fertilizers is prominently because it is eco-friendly, does not causes pollution in the field. Okay, when you are using microbes as a biofertilizer, you are inhibiting, you are avoiding pollution, soil pollution, air pollution, water pollution. Okay, and simultaneously the age of the soil is also increased. Okay, bacteria, fungus, cyanobacteria are the major source of biofertilizers. Okay, so first we have bacteria as a biofertilizer. The nodule on the roots of leguminous plants are formed due to the symbiotic association with rhizobium. Rhizobium is an bacterium. Okay, it is an heterotrophic bacteria. Okay, which lives in the nodules, root nodules of leguminous plants like your pea. Okay, gram, pulses. Okay, Leguminaceae family, Fabaceae family. These plants root contain nodulated roots, and in that nodulated roots you will find a bacterium which is rhizobium, and it is live in symbiotic relationship with the plant to do and produce nitrogen. Okay. Some bacteria like Azotobacter bacteria is another example of biofertilizers. Okay, unlike rhizobium, these are not symbiotic. Okay, your Azotobacter is not a symbiotic. Okay, let's see. So we have symbiotic or non-symbiotic bacteria. Okay, symbiotic bacteria which you find inside the fields, and non-symbiotic bacteria also they are inside the field, which do not live in a relationship. They are doing free nitrogen, that free life cycling and free fixing of nitrogen inside the field. Okay, so your symbiotic bacteria like examples are rhizobium. Okay, rhizobium, which found in legumes plant. Okay, second example is this first. Second example is your Frankia. Okay, it will present in the roots of ulnus, again in leguminous plant. Okay, these are symbiotic. Okay, Anabina in azolla leaf. This is an BGA. Okay, Anabina is a BGA, blue green algae or your cyanobacteria. So these all are symbiotic. Rhizobium, Frankia, Anabina, these all are symbiotic. They are live in a symbiotic relationship with plants. Now we have some non-symbiotic which lives free living. They are not associated plant, but they are present inside the soil and they doing their nitrogen fixation. They are not dependent on anything. They are doing their thing freely. They do not require anything. They are free. They are single. Okay. So example is your Azotobacter, Azarospirillum. Okay, Azotobacter, Azarospirillum. These are non-free. These are free living bacteria. They are not dependent on anything. They are non-symbiotic bacteria. Okay. All these bacteria play an important role in nitrogen cycle. Okay, as I said, these are absorbing nitrogen. They are making nitrogen. They are doing nitrification, denitrification. They are running nitrogen cycle inside the soil. So these are very important for the absorption of nitrogen for plants. The excessive nitrogen which plant cannot produce, these are aided inside the plant body by the help of these bacteria. Okay, they fix atmospheric nitrogen and form ammonium. Okay, how they will help the plant? They take nitrogen from the atmosphere and convert that nitrogen into ammonia. Okay, later this ammonium is converted to the compound of nitrogen and used up by the plants. Okay, now after this ammonium ions is converted into nitrogen compound like ammonia and other substances which is uptaken by your plants. Okay, this way they are bacteria enhance the nutrient quality of the soil and act as a biofertilizer. That's how they act and enhance the quality of soil and act as a biofertilizer. Okay, you can see here this is atmospheric nitrogen absorbing by your rhizobium and azotobacter and converted into organic compounds. Okay, and which is absorbed by your leguminous plants. Okay, next we have fungi as a biofertilizer. That is your glomus genus fungus. Okay, glomus, you studied that thing in the form of mycorrhiza in 11th class in your biological classification. Okay, so mycorrhiza symbiotic relationship between fungus and roots of higher plant, mostly pinus. Okay, here the fungi absorb phosphorus. Now remember, previously we studied about nitrogen fixer organisms. Now we are dealing with the organism which do the fixing of phosphorus. Okay, phosphorus is fixed by a fungus of glomus genus. Okay, here the fungus absorb phosphorus from the soil and pass into the plant body. Most of the fungi that belongs to the genus glomus form this mycorrhizal relationship. The relationship between the roots of higher plant and glomus genus fungus is called mycorrhiza. Okay, in which that fungus is giving and absorbing phosphorus for the plant while your plant giving nutrition and home shelter to that fungus. Okay, then we have cyanobacteria as biofertilizer. Okay, your BGA, blue green algae, the bacteria who can perform photosynthetic respiration. Okay, or you can say they will produce a photosynthesis process. Okay, autotrophic organism they are, they can produce their food because they have the power to do photosynthesis. They will produce oxygenic photosynthesis in which they produce oxygen. Okay, so these are the cyanobacteria as a biofertilizers. Okay, so cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environment. You will find cyanobacteria in water and in soil also. Okay, these are autotrophs. They produce their food their own. Previously that rhizobium, your Frankia, your azotobacter, your azotospirillum, these all are heterotrophic bacteria but they do nitrogen fixation. Okay, but your cyanobacteria is an autotroph bacteria which can produce its food itself and it is an also a nitrogen fixation organism or your biofertilizer. Okay, many of which can fix atmospheric nitrogen. Example, Anabina, Nostoc, Ocetuloria. Okay, these all are the example of your BGAs which do nitrogen fixation, which act as a biofertilizer. Again I am saying the name, Anabina, Nostoc, Ocetuloria. Okay, Anabina azole is the most famous nitrogen fixer BGA. Okay, again I am saying the name, Anabina azole is the most famous, okay, and most common that nitrogen fixation BGA. Okay, is a cyanobacteria which is lives in symbiotic association with free floating water fern, that is a pteridophyte, azola. Okay, azola is the smallest pteridophyte of earth and it have a symbiotic relationship with your that Anabina BGA. Okay, and the species of Anabina that is making that symbiotic relationship with azola is Anabina azole. Okay, Anabina azole fixes atmospheric nitrogen and forms organic compounds similar to the bacteria. Okay, the organic compound which you studied in bacteria photo, that bacteria biofertilizer part, okay, that is ammonia. Okay, all these are also produced by these symbiotic BGAs. Okay, this is your Anabina azole and this is your Nostoc. Both are fixed atmospheric nitrogen, both do nitrogen fixation. Okay, Cyanobacteria in paddy fields. Okay, paddy fields means rice fields. Okay, so in paddy fields, cyanobacteria serve as an important biofertilizer. Blue-green algae also add organic matter to the soil to increase the fertility. Here you can say this green. This is not soil. This is not grass. This is water which containing BGA. Okay, cyanobacteria in paddy field. These all are cyanobacteria. This is middle line is your paddy field, paddy crop. Okay, rice crop. And the water you are seeing here is fully occupied by BGA. Okay, cyanobacteria aids organic biomass to the soil. Okay, you can see Ocytoloria. Okay, and that Anabina. Okay, sometimes that Frankia also present inside these paddy fields. Okay. Currently, a number of biofertilizers are available commercially in the market, and farmers use these regularly in this field, replenish soil nutrient and to reduce dependence on chemical fertilizer. You can go in market and you can buy a biofertilizer. Okay, it is less in cost than your chemical fertilizer. And second thing, it is reducing the pollution or does not do the pollution in the atmosphere, in the soil, in the water. It is a clear fertilizer. Okay, so these all are different commercially available biofertilizer. This is rhizobium. Okay, nitrogen fixing bacteria. This is your phosphate solubilizing bacteria, PSB. Okay, and this is your azotobacter, azovita. Okay, these all are different biofertilizer which you can get inside the market, and they do not disturb the ecosystem. They do not cause any pollution inside the ecosystem. Okay. So now we are going with a summary what we studied today. First we studied about biogas production. Now slurry tank when that slurry tank is emptied inside the digester, it produced by anaerobic respiration your biogas, and this biogas get out from that outlet in the form of that gas and transported to different area. Now the remaining part that goes into the sludge tank and it is used as a fertilizer. Okay, biocontrol agent we studied about virus which is a baculovirus. Okay, then a bacteria that is Bacillus thuringiensis, then fungi trichoderma, and animals like ladybird and dragonfly which is used as a biocontrol agent, which is used as a biopesticide. Okay, next we studied about biofertilizers. So we have two, bacteria and fungus, which lives in symbiotic relationship with the leguminous plants, and we have a free living bacteria also which does not live in any relationship like azospirillum and azotobacter. Next we studied about a fungus which live in a symbiotic relationship with the roots of higher plants. That fungus is your glomus, and the relationship is called mycorrhizal relationship. Okay, forms mycorrhiza genus fungus. Okay, so now this is the end of the third part of your microbes in human welfare, that is production of biogas and that your biocontrol and biofertilizer. In next class we are going to study about microbes which is used in wastewater management sewage treatment plant. Okay, in which we studied primary and secondary treatment. I have one video also which I will show you that can easily understand you what is these primary and secondary treatment. Okay, for now thank you for the watching the video. Like, share, subscribe and kabhi jhukne ka nahi.